Hello, my name is Lauren Ashcraft, and I'm thrilled to welcome you to our biannual research webinar for our valued donors. You play a critical role in our mission, and today we get to hear directly from an American Cancer Society research professor, as well as a metastatic breast cancer patient, about how your support of research helps make incredible breakthroughs possible. At the end of our presentation, we'll be answering a few questions. If you would like to ask a question to our presenters, please press *3 if you've joined us by phone. If you're listening through your computer, please use the chat box to submit your question. You may also use this chat box should you experience any technical difficulties during the webinar. And now, it is my pleasure to introduce our first speaker and moderator, Dr. Susanna Greer. Dr. Greer joined the American Cancer Society as Scientific Director of Clinical Cancer Research and Immunology in 2015. She received her PhD Immunology from the University of Alabama at Birmingham. She was a postdoctoral fellow at the Lineberger Comprehensive Cancer Center at the University of North Carolina, and she was recruited to Georgia State University as a Georgia Cancer Coalition Distinguished Cancer Scholar. To support scientists in developing skills in outreach and communication, Dr. Greer developed the internationally offered course, The Art of Science Communication. She served as a science communication consultant, and she is the co-host of the American Cancer Society's research podcast, Theory Lab. Now, without further ado, here is Dr. Greer.
Thank you, Lauren, and hello, everyone. I'm so excited for this opportunity. We are going to have such a great presentation today and such a nice opportunity to ask some questions. So thank you for joining us for this ACS research webinar. All right, so what are we in store for? Well, today we're going to learn about both the science of cancer metastasis and the pursuit of more effective therapies. And we are also going to have a really wonderful opportunity to hear from a metastatic breast cancer thriver who is going to share her cancer story with us and her cancer advocacy journey. So before we get started, I think we should just level set that we are living through what can really only be described as an unprecedented time. But I am just really encouraged to see so many people joining the webinar and such great support for the American Cancer Society and for our effort to advance cancer research. So thank you so much for being with us, and it just demonstrates to all of us that we can continue to be together even when we're apart. And we've all come today for different reasons. We're all advocates in support of research. We're supporting those that we've loved and lost to cancer, and we certainly are supporting each other. And as Lauren indicated, we are just honored today to have two special guests with us. So I'm going to introduce them first, and then we're going to hear from our speakers, and then we'll have an opportunity to ask them questions. So joining us today is American Cancer Society Research Professor, Dr. Ribbon Kong. Dr. Kong is a Warner Lambert Park Davis Professor of Molecular Biology at Princeton University and an Associate Director of Rutgers Cancer Institute of New Jersey. Dr. Kong graduated with a bachelor's degree from Fudan University in Shanghai in 1995, and after completing his graduate study at Duke and postdoctoral training at the Memorial Sloan Kettering Cancer Center, Dr. Kong joined the faculty of Princeton University as an assistant professor of molecular biology. He was promoted to associate professor with tenure and to endowed chair full professor in 2012. Dr. Kong's research focuses on the mechanisms of breast cancer metastasis, and his amazing work has discovered new genes that promote cancer recurrence and metastasis and chemoresistance of breast cancer. And he's helped us to understand the interactions that are so important for metastatic growth. Dr. Kong is an individual that the ACS recognized early on was just going to be fantastic. He received an ACS Research Scholar Award in 2005 and was selected as an ACS Research Professor in 2019. So welcome, Dr. Kong. We're so glad to have you. Also joining us today is my wonderful colleague, Rachel Fournier. Rachel was diagnosed with de novo metastatic breast cancer in 2015, and at the age of 38, with no family history of cancer. Rachel has advocated for funding for cancer research at the federal level and has served as a patient advisor for the development of several new breast cancer drugs. She has participated on many conference panels to share her experience of living joyfully alongside cancer, and she takes time to counsel newly diagnosed metastatic patients. Lucky for all of us, Rachel joined the American Cancer Society in February 2020, where she is the Director of Foundation Relations for the Northeast region. In her work, she hopes to do her part to advance life-saving cancer research and provide access to care for those most in need. Rachel lives outside of Philadelphia with her husband, her teenage daughter and son, and a rescue dog. Welcome, Rachel. We're so glad to have you. And Rachel, I think we'd love to start with you. Would you be willing to share your story with us?
Yes, thank you, Dr. Greer. Thanks so much for the invitation, and I'm really delighted to share just a little bit of background on what my metastatic breast cancer experience has been and how it really, links with the advocacy work and the need for research and the amazing work that Dr. Kang is doing through his ACS professorship. So if we sort of harken back to where my cancer experience began, in April of 2015, I was working in a field that I loved. I was in a brand new job. I was 38. I had no history of cancer in my family. I was too young for a screening mammogram. I had been married for quite a long time to the boy that I met at summer camp who became my husband. I had an 8 and a 10-year-old, and we were really, living our best lives. We were on vacation frequently. We just had this really, really wonderful life. And in April of that year of 2015, we went on vacation and went jet skiing for the first time. And over the course of the week, I started having really nagging back pain that I had never had before. And so as soon as I got home, I went to urgent care, who did an x-ray and didn't see anything or any damage. And I followed up with my primary care physician who said, you're almost 40, so of course you have back pain. That's to be expected. And I'm going to refer you to physical therapy, and we can sort of see if we can strengthen the muscles in your back and hope that this is just something that we can, kind of monitor, but certainly nothing to be worried about. And so over the course of that summer, the back pain just became more and more excruciating. And as I went through physical therapy, I was becoming more and more debilitated to the point that I couldn't walk. I mean, I got to the point of really just losing most independence. And so I finally was able to get an MRI, and the MRI came back that I had fractures in 3 places in my spine. And, my first thought was, oh my gosh, that's so embarrassing. Like I'm in this new job. I might have to have surgery or wear a back brace, and this is going to be a really inconvenient, few weeks. And I went to see the orthopedic surgeon for the first time, and thinking it was something that was easily fixable, and I was 38 and healthy, and what a fluke that I had this back pain. And his face really just fell, and he said, in like a really paternal, caring way, you have 3 spinal compression fractures, and what that means is that cancer has eaten away your bone, and the vertebrae are virtually non-existent. Your spine has sort of collapsed upon itself. And not only that, but in the bone scan that he did, he said, you have cancer in about 40 different places in your skeleton. And I've only ever seen this in someone as young as you with a metastatic breast cancer diagnosis. And I think I certainly was stunned and devastated. And, I immediately went home and went on cancer.org and looked up what even is metastatic breast cancer. What does that mean? And what I immediately found out was that metastatic breast cancer has a 28% 5-year survival rate. So I went from 38 and thinking, my grandmother is 95. So, thinking I had this really long healthy life ahead of me to suddenly be measuring things in, if I am really lucky, I will have 5 years. And that's just devastating. And so, life stopped, and I came home and I couldn't look in the faces of my children. My husband and I were terrified, and we went to the first meeting with my medical oncologist, and, she confirmed the diagnosis and shared, yes, this is incurable, and you will be in treatment forever. And she wrote down on this index card, here are the treatments that are available to you, and what will happen is, you'll go on one treatment and we'll hope to get you, months on that treatment, and when that one stops working, you'll move on to the next treatment, and the goal is that, you keep having another option. And that a list that she handed me was really short, and really finite, and, everything just sort of stopped and froze in that room, and, you're focused on everything that you're about to lose. And, for me, I felt such ownership of being about to destroy my family with this diagnosis. And, we sort of sat in silence for what felt like hours. It was probably 45 seconds. And she said, but then she said, while you are progressing through this list, there are researchers working in labs. There are patients in clinical trials. There are pending FDA approvals, and our goal is that the list will continue to grow so that you will never run out of options, so that when you are at the bottom of this list, we've created a new list. And that offered so much hope and, allowed me to dare to even think of what a future could look like, sort of living alongside metastatic breast cancer. And it just gave me so much gratitude to researchers like Dr. Kang. And, when I go to treatment at the facility that I'm cared for, I always kind of look up in the research tower and, sort of send my good wishes and all my hopes and thoughts to the researchers that are working there, because I know that their work, will prolong my life. And Dr. Kang and I had the opportunity several years ago to be in D.C. together, both hailing from the great state of New Jersey, and visiting on the Capitol and really speaking to the need for research and the need for research funding so that my list will not run out of options. And so I'm just so grateful to be able to, contribute in a small way to this dialogue, but I'm especially grateful for, the support of research like Dr. Kang's that you all have invested in, and I'm really excited to hear along with you an update on his work.
Oh, thank you, Rachel. Goodness, what a beautiful story. And I think as you were speaking, I was just watching the mental image in my mind of you living your best life, and then your journey to urgent care and physical therapy through debilitating pain, and finally just this crushing diagnosis, all the way to what you shared with us, which just, it's so uplifting that your goal is that list continues to grow and your treatment options never run out. And that's certainly our goal for you. And you're exactly right. It's because of wonderful researchers like Dr. Kang that we have these opportunities, and wonderful donors like all of you on the phone. So, Dr. Kang, if you're ready, we would love to hear about your research.
Great. Can you hear me?
Yes, absolutely.
Okay, good. And can you see the slide?
Yes, we sure can.
Okay, good. Well, thank you, Rachel. It's such a touching story, and it really is a great reminder about why we work so hard in the lab. When Princeton University come back and reopen after the initial wave of COVID-19, the first thing that we open is research, because that is so essential, important, not only for our students, but also for the mission of the university. So when I Can you see the slides of the Fortune magazine on the screen now?
Yes.
Okay, great. I just want to make sure the advancing of slides is working okay. I started my lab in Princeton University in 2004. And I remember one day I was flying for my interviews in different universities. I paid attention to a cover of the Fortune magazine. And on it, it says, "Why are we losing the war on cancer?" And the main conclusion is that because we don't understand how metastasis happen and we do not know how to stop it or reverse it. So that was in 2004. And when you look at the next slide, looking at the survival of the patients after initial diagnosis, you can see that since 2004 and even up to 2012, the survival of patients who are diagnosed with early disease in different kind of disease, especially breast, prostate, and other cancers, the orange line is approaching 100%, and even for some more aggressive cancers, lung cancer, the survival rate is not too bad. But for patients who have metastatic cancer at diagnosis in red line, the survival rate is much lower, and also it doesn't really change much since 2004. So that still call out for the question of how we are doing in terms of treating patients with metastatic disease. There are some progress being made. For example, when you look at the third graph for melanoma, you can see there is an uptick for those stage 4 metastatic melanoma, and that is in part because of the breakthrough in immunotherapy. So the bottom line is that metastasis is still the major cause of death among all major solid tumors. And we still need to grow the list of medicine available for Rachel and other people so that they always have another option to deal with the disease when the list is coming shorter. And what I'm going to share you today is some of the work that we did in my lab that was supported by American Cancer Society that hopefully will lead to the growth of that list of medicine for metastatic disease. So what you're looking at here is a article that we published about 10 years ago. That is a landmark study trying to figure out why in certain breast cancer patients, even when they are diagnosed with early stage disease, they are likely to come back with recurrent disease that are metastatic and resistant to treatment, the so-called poor prognosis cancer, breast cancer. And what we did in that study is we used genomic tools and we used complicated computational approach and eventually identify a gene called MTDH, met adhering. The protein, as you can see in here, is shown in yellow color here. That is a piece of the protein that binds to its partner called SND1 in green. And you may not understand what does it mean in terms of this binding, but the bottom line is the importance of this interaction allow cancer to become highly metastatic and also be resistant to treatment. But because we have this high resolution understanding about this so-called crystal structure of the molecular interaction between MTDH in yellow and SND1 in green, we figure out that it's a key interaction. And I try to use the analogy of if you go bowling, you use your fingers to hold on to the bowling ball. And what you can see in here are two red so-called amino acids residues that are like fingers holding into the two cyan color pockets in the surface of this green SND1 protein. And this interaction allows the proteins to function and drive the aggressiveness of cancer. So if you are a drug company executive, it will come as an immediate thought that one way to develop medicine is to come up with chemical compounds that basically insert into those pockets and do not allow the finger to hold on to the bowling ball anymore. And that disrupt the function of the protein and make the cancer much more curable. And this is exactly what we did. The science or intellectual property discovered at Princeton University is now licensed to a biotech company, and the biotech company have come up with a compound that blocks this interaction and hopefully is moving on to trial in the patients. At least in the mouse model, we know that the compound works well, reduce the recurrence, reduce the metastasis, and make the cancer much more sensitive to chemotherapy and immunotherapy. Another example is actually related to Rachel's condition, which is bone metastasis. About 70% of stage 4 breast cancer patients have bone so-called skeletal complications, bone pain, fracture, hypocalcemia, and so on. And we currently have a couple of medicines, denosumab and bisphosphonates, for treatment of bone metastasis, but those do not provide cure. What we discover in another study that you're showing here is we again discover a interaction between the tumor cell, shown as the cartoon in blue here, and how they cross-communicate with bone cells in pink color and brown color. That causes the bone destruction, because bone is actually not destroyed by tumors. It's destroyed by the so-called osteoclast, the bone resorbing cell in our healthy body that become hyperactive and become slave labor for tumors to break down the bone, for tumor to grow. And this study led to the discovery of key molecular interactions between bone cell and tumor cell. And we work with a drug company called Amgen. Somebody probably heard about the company because they developed denosumab and many other medicines. And we come up with an antibody called JAG1 neutralizing antibody. And we are currently developing that for making bone metastasis treatment much more effective. You can see that in the mouse. In the left is the untreated mice, and those colors are showing development of bone metastasis throughout the body of mice. In the right side is the mice that are treated with combination of chemotherapy and antibody, and it's almost completely free of bone metastasis. So it is really encouraging result for us. Another area that the lab is getting into is the area of immunotherapy. We know some of the patients respond very well to immunotherapy, but some of the patients don't respond at all. And part of the reason is the tumor developed resistance to the immune cell killing or evade the immune cell attack. And in another study that we published a couple of years ago, we showed that the so-called cancer stem cell, the kind of root of all cancer, the root of cancer recurrence and metastasis, is in part because they express a particular microRNA, small molecular RNA called microRNA 199A, and that makes the cancer stem cell particularly resistant to immune cell attack. And by discovering this new information, we hopefully will make the immunotherapy more effective for the patients. And finally, the last example, again, was just published last year in the lab, again, dealing with the condition of bone metastasis. And in this particular case, we are looking into how we can prevent patients from getting bone metastasis after their initial diagnosis of early stage cancer, because a lot of the patients are diagnosed with early stage disease, get surgery, get chemo, and they thought they are free of cancer, but unfortunately, a few years or even a couple of decades later, they come back with a bone pain and a bone metastasis develop. So the tumor cell somehow can remain dormant in certain part of the body and then wake up one day and develop a metastasis. And we have tried to look into how do those tumor cells remain dormant and how do they become activated at certain conditions. And what we found is that in the bone, when tumor become bone metastasis and tumor cells here are shown in green color here, they seem to be hugging the blood vessels in the bone, the so-called vascular niche that protect the cancer cell and also make them able to grow to become bone metastasis. And again, what we discover is a key molecular interaction called E-selectin is a molecule expressed in the blood vessels, and it's like glue or Velcro that allow the tumor cell to attach to them and gain aggressiveness, gain the so-called cancer stem cell activity, and then grow to become bone metastasis. The good news is that this E-selectin actually has been a target of therapeutic development for another unrelated condition called sickle cell anemia. And so there's a compound called is a compound that looks like sugar. It's a sugar analog called glycomimetic compound that has been shown to be able to block E-selectin function, because E-selectin binds to its partner through molecules that look like sugar. And so this glycomimetic compound trick the tumor cell to bind to them and not able to bind to the blood vessel, and that reduce the recurrence of bone metastasis. And because also the compound is just like a sugar, it's really not doing harm to the body of patients. It's unlike chemotherapy. And we hope that maybe by combining this glycomimetic compound with chemotherapy, we could reduce the risk of patients developing metastasis, bone metastasis later. So the work I'm show to you, you are actually involved in all of this because of your support for ACS. I was able to be funded by ACS in basically the first year of my independent career as a cancer researcher at Princeton University. So I received the Research Scholar Award in 2005. That's when I get started. And the discovery of MTDH is funded by that initial American Cancer Society Research Scholar Award. My recent selection as the American Cancer Society Research Professor have a goal to support my research in the area of bone metastasis. And this is the area that we have made a lot of progress, and we are also hoping to continue to develop new treatments based on discoveries made in the lab. The goal, as Rachel said, is to continue to grow the list of the medicine available for stage 4 patients, and in a speed that actually will outpace how the cancer come up with new ways to evade the therapy. And hopefully even better is one day, maybe just like immunotherapy for melanoma and some lung cancer, we could come up with a very effective combination of treatments that not only will slow down or block the continued expansion of metastatic cancer, but actually completely reverse it and eliminate metastatic cancer from the patient's body and make them live free without the worry of the disease anymore. That's our ultimate goal. And with that, I want to show you the picture that Rachel and I actually took together in front of Cory Booker's office in the Capitol Hill. We work as partners, hand in hand, the researchers and advocates, to educate our lawmakers the importance of continued federal support for cancer research, and also the importance of changing legislatures that support the care and financial well-being of cancer patients. So with that, I want to end my portion of the talk, and I'll be happy to answer some of the questions later.
Thank you so much, Dr. Kong. That was just fascinating, and we're also excited and impressed. All right, we have lots of questions, so we'll get started. I think it might help our audience to understand a little bit about starting research in a mouse model. So maybe I can ask this question in two parts. The first would be, why would you start in a mouse? And the second would be, can you walk us through how do you actually move research that's done in mouse models to where it impacts humans?
Right. So why we start research in mouse? There are many considerations. One is that you cannot do a lot of research in the patients because of the ethical reasons and also safety for the patient. So when you apply medicine, even running clinical trial in the patients, you have to make sure that you've done everything possible to minimize the risk to the patients, even if it's an experimental medicine. And animal models provide a great platform to test that. Of course, when we do animal research, we also follow very strict federal guidelines for the care and well-being of animals. We try to reduce the number of animals as much as possible because we don't want unnecessary sacrifice of animals. And we also follow very strict guidelines in terms of, for example, statistical considerations. Again, that is to reduce the minimum number of animal use. And also we try our best to minimize the pain to the animals when they develop metastatic cancer. But the mouse, and also there are a few other rodent models and some the model use other vertebrate animals. The mouse model is a great model because they are mammals. And for example, for breast cancer, like humans, they also have mammary glands. But there are some advantages there. One is that they have a relatively short life cycle. and so we can do many of the genetic study relatively quickly. That will accelerate research. And also, we know the biology of mice really well compared to humans, and their genes and their genomes have a lot of similarities with the humans. Like humans, they also have, many of them have the intact immune system. Some of them don't have the intact immune system. They are immune deficient because in those models, we can transplant human tumor into mice, and that will be an even closer mimicry of the disease in the patient. So, for example, there are so-called patient-derived xenograft model in which you get the tumors from patients and then directly transplant into immunodeficient mice. And you can evaluate how tumor grow. You can evaluate the treatment of experimental medicine. So, so animal models, especially mouse models, have been great tools, and science has advanced to a point that not only we can try experimental treatment in the mice, we can also do genetic engineering of mice so that we can, for example, specifically delete certain genes that we think is important for cancer and use a genetic approach to show this is a good target or not. Two criteria. One is if you eliminate this gene, does the tumor shrink? And second is if you eliminate this gene in the mice, does the mice suffer? Is there any side effects? So that provides really key information about whether it's an effective target and also whether it's a safe target. So that hopefully answers the question about why we started research in the mouse model. And the second part of the question is about how do we move from there? It's a great question. So there are many steps that will need to go through to go from an animal study to eventually a medicine in the patient. So when we initially, the discovery is almost always made in academic research lab at universities. And usually it will be a discovery of a gene or a pathway that is important for a certain aspect of cancer. And usually such finding will be published in a scientific journal. It will be shared in the community, and then everyone understands this could be an interesting target to develop potential medicine. So some of the university lab and sometimes a biotech company and sometimes a drug company will try to do drug screens. For example, as I showed you earlier, try to find a compound that binds to the keyhole of that bowling ball of the protein. And it's so-called high throughput screening. So the companies will go through a screening platform to screen oftentimes millions of compounds, chemical compounds. Some of them exist in nature. Some of them are chemically synthesized by chemists. And after you go through millions of them, you will find a few hits. But those hits are usually not medicine, because either they are not very soluble or they have terrible, they don't have good ability to penetrate the cell membrane to go into the cancer cell, or they cause toxicity in the patients. Certain things that don't make them great medicine, but it's a start. You have a hit molecule, and then the chemist will come in. They will use their brain to design chemicals that look like the initial hit, but a little bit different to solve some of the solubility or toxicity or permeability problems. And then you go from a hit to a lead compound that look very promising, have therapeutic effect in the animal models, have very, very little toxicity, and you could take it orally and has great solubility in the blood. Now you go from there, you do all the testing about those pharmacological profile, safety in the animals, and then you go to the FDA and you file the so-called investigative new drug, IND filing. That will then start to give you the permission to design phase 1 clinical trial. Phase 1 is to test the safety in the patients. Then phase 2 and 3 will be testing them in growing number of patients for their efficacy. And then if the results are great, then we move on to get approval for treating them in the patients. This is the approach. And usually it takes, in the past it probably takes 10, 15 years. And now there are, probably some better ways to accelerate some of the process. Some of the medicine could go from discovery to approval in a matter of even like 5 years or shorter.
Wow. Okay. So you've helped us to understand exactly why you use mouse models. We have a lot to be thankful for mice, and how the trajectory, this long path of publishing findings and collaborating with drug companies, moving through safety and FDA regulatory trials, to where we actually have a new therapeutic in the clinic. So we've had another question come in that's closely related. So maybe could you help us understand, for your research specifically, and you talked about lots of different areas of research, and maybe you talked about bone metastasis and immunotherapy, you talked about the vascular therapeutics that you have, but we've had a question come in about how close do you think your findings are to clinical trials and new therapies? I think our audience would love to hear that.
Yes, great. So, so they are medicine. Some of the compounds that I mentioned earlier are in different stages of this pathway that I just mentioned. For example, the glycomimetic compound that is developed by a biotech company called Glycomimetic. It was actually listed in NASDAQ and so on. So they actually already have a compound that I think passed the FDA approval for sickle cell anemia. And so there's a possibility of repurposing that for the treatment or prevention of bone metastasis. But because it's a different indication, the clinical trial would need to be run and designed specifically for either treating or preventing bone metastasis. For treating bone metastasis, probably it will be a shorter trial because it will be patients with existing conditions and then treated with this compound. Maybe it will combine with standard of care and see if they have less bone pain or less complications. But it's a relatively shorter trial time window. Prevention trial is harder because you have to recruit a larger number of patients, early stage patients, and because only a small number of them will develop recurrence to bone. And you also have to wait much longer time because it takes time for them to have recurrence. So usually those are harder to do because of one is the time and second is the financial cost is higher. When you run a trial longer with more patients, it increases the cost significantly, and the company usually has to consider that. So then it will be the decision of the company to evaluate whether they have enough financial means to run one or another type of trial in what cancer that they have a clear indication of endpoint, and then it's a matter of maybe 4 to 5 years to get the approval to do phase. Well, they don't need to really do a lot of phase 1 because it's already approved for a different disease. But at least phase 2, 3, it will take time to finish that. So that is the compound that related to the vascular blood vessel niche. The other compound that I mentioned about bone metastasis is this the crosstalk between tumor cell and the bone cell, mediated through a cell surface molecule called JAG1. And I mentioned that the drug company, which is a much bigger drug company called Amgen, have a compound for blocking JAG1. Again, that is a compound that has been humanized, complete human. It is actually a human antibody that developed in mice, but the mice is humanized, so they produce human antibody, and you can go to the patient directly. That antibody has already done some pharmacological characterizations. As far as I know, the company is also considering that antibody for another condition called COPD. It's a lung condition that causes also a lot of deaths in patients. So again, it's a decision of the company to decide whether they want to go with COPD first or go together with bone metastasis treatment first. And there are actually a lot of considerations about not just, I want to treat the disease, but also about what if the trial fails in one disease, does it affect the trial of another disease? And what is the financial consequence of that? So it's a very complicated calculation that the drug company has to do. And then once they're committed to it, again, it will take probably 4 to 5 years to run an appropriate trial in the patients with bone metastasis or to prevent patients with bone metastasis. But both cases are later case candidates that are already more mature in terms of molecular characteristics And so the pathway toward approval or using the patient, once the company is committed to that path, should be relatively quick. I'm saying maybe about 4 to 5 years in an ideal situation. The first example I gave about that bowling ball example, that's the early stage. It was actually been done in my lab. We use that as a target. We screen the compound. We screened 50,000 compound in Princeton University, the chemistry department. We found a hit. The hit looked promising, but it does have a downside, which is you need to have relatively high concentration to inhibit the protein interaction. And so we need to find a better compound. So as you can see, this is a earlier phase in terms of the step of drug development. We have a hit, but we don't have a lead yet. This is a hardest part from hit to lead because it's the so-called zone of death in drug development. Is you could have a great target, you could have a hit, but you may or may not be able to eventually get a great lead to go into IND and eventually, testing in the patients. But the target, we do have strong belief that it's a great target. So we are not banking all our hope in one compound, right? So we are screening more compounds. We are also trying to find a different approach. Instead of blocking the protein, we could use so-called antisense oligos to block the gene directly. And so we are coming up with plan B, plan C, and plan D in case one fails, the other one may work. And that will probably take longer. I'm expecting more like 5 to 10 years for development. And now we do have a chemical compound in the hit stage. We have an antisense oligo that is also working. Again, it's an initial hit, but it's a different pathway toward approval. So these are actually good examples to show you that it takes tremendous efforts to bring one, out of any initial hits, probably only less than 1 to 5% of them eventually make it to as a medicine. And each one of them costs billions of dollars to develop. So it's a huge enterprise and effort, and it's a coordination between basic scientists in universities and drug developers in pharmaceutical industry. But we all have a same common mission, which is to bring new medicine, grow the list for the patients.
Dr. Khan, I think you're exactly right. It's so interesting what you shared with us because you've helped us to understand how basic science moves to animal models and then drug screening and trials, and you're exactly right to grow those options on Rachel's list. So I think I'm going to answer one question really quickly myself, and then we have time for one more question that kind of directly ties into that. We have a question that's near and dear to our heart at the American Cancer Society. How many lives are saved from breast cancer today? It's a tough question, but we know that American Cancer Society research has shown that the death rate from breast cancer dropped 40% since 1989, so until 2017. So, Dr. Khan, we are grateful to you and your colleagues and everybody who plays a role along that process, including, of course, the donors that are on the phone. I think we have time for one more question, and then I'm going to, after we hear from you, I'd love to hear from Rachel to see if after hearing all of this, what she'd like to share. But this last question is, I think, really helps tie up everything that you have shared with us, all the different avenues of research that you're approaching, and that is that cancer isn't just one entity, right? There are hundreds of different cancers, and even within cancer subtypes, there are different subtypes. So can you help us to understand how we determine which patients might respond to a particular therapy? And maybe you could put it in that process that you just described to us as we move from basic science to mouse models to collaborations with industry and then to clinical trials. Where do we begin to understand the impact that a new drug will have on that inline cancer patient?
Yeah, that's, again, a great question because when we talk about cancer, it's actually not one disease, it's hundreds of different diseases. And even just for breast cancer, there are probably, 20 different subtypes, and each patient is unique. And I cannot emphasize enough that, so-called precision medicine approach is really key to the success of the treatment for any given patient. A drug may work wonderfully for one patient and may not work at all. And when a drug fails, we lose critical window for the patient, and the patient's condition might deteriorate. That might prevent the uses for other drugs. So indeed, it's a key question about how do we know if a drug is going to work or not for this patient. And it's a fundamentally important. Actually, many of the drug development nowadays, when FDA is providing approval, one of the criteria or common question asked is the so-called biomarkers, that so-called companion diagnostics, that is useful to identify in patients who are likely to respond and benefit from the treatment. And I will give you a couple of examples. For patients who have HER2 positivity, HER2 amplification, they will respond very well to Herceptin. And if you apply the treatment to patients who are negative for HER2, they are much less likely, almost unlikely to respond. And same is anti-hormonal therapy. If the patient is negative for ER, they are unlikely to respond to tamoxifen and other anti-hormonal therapies. So almost nowadays, every single targeted therapeutics has to have a companion diagnostic, because that's how we select patients, not only for treatment later, but also even for the initial clinical trial. Because imagine if the Herceptin trial is done in all breast cancer patients, it might actually fail the trial and never reach the market, reach the patients, because only a small fraction of them are HER2 positive patients, and only those patients will respond. So you'll get a so-called false negative result if you take all the patients into consideration. So of all the medicines that we are trying to develop now in the lab and in the pharmaceutical company, we always, with the goal of finding the key drivers of this aggressiveness of cancer, we know in each of the slides I show we have a specific target. So for those patients, we have to treat them only if they have a diagnostic test to confirm that they do have a high-level expression of that particular target. This is also true for the current immunotherapy. Before the patients receive the immunotherapy, the doctors will want to have their PD-1 tested and other markers tested to see if they are a good candidate for that treatment. This is not to say that, well, we don't want to waste our medicine. It's actually more important to say that we don't want to waste the opportunity to have a effective treatment. We don't want to apply a non-effective treatment that causes you to lose the time and process, good condition for treatment. So, that's one point. The other one is that even though cancers are very diverse, one cancer is very different from another, there are emerging common things among different cancers. And we start to see the blurring of lines. For example, we can treat breast cancer patients and ovarian cancer patients, and even some of the prostate cancer patients with the same kind of medicine that if they are deficient for the breast cancer gene BRCA1. So again, this is not just treating breast cancer, it's actually treating cancer that are deficient in BRCA1, a tumor suppressor. And many of the genes that I'm telling you today, MTGH, we initially discovered in breast cancer, and later on we show it's equally important for prostate cancer, for lung cancer, for colorectal cancer. So cancers are diverse, the mechanisms are diverse, but they are also common themes. And that's the hope, that we'll find the common themes among cancer. And we are going to be outsmarting cancer in finding out the smart bullets, or to solve the enigma of cancer with the right cocktail of medicine.
Well, thank you, Dr. Kang. That was really helpful for us to understand how do we determine which patients are most likely to be impacted, and helps us to understand the process all the more better. I'm going to take the one last question for myself, and because I talk to cancer researchers all over the country all day long, and there's a really wonderful question that relates to COVID. So along with new revenue, what will it take to relaunch research efforts in the wake of the pandemic? And I do want to share just a little bit of positive news there and hope, and that is that, and I imagine it's true for you as well, Dr. Khan, there is a silver lining to the pandemic in this space, and that is that while we have certainly research has been and still is impacted by an inability in some institutions still to be in labs, or at least for labs to be fully staffed, the silver lining is that scientists are, in the face of COVID, learning brand new ways to interact with each other, to have international virtual collaborations, to attend virtual meetings. And probably one of the best things that I have heard is that researchers are having to make decisions about, we can't do everything. What is the most important question that we need to be asking as a researcher right now to make the biggest impact? And so they are taking this time to read, to reflect, to write, so that as labs fully open back up, they will be able to make the best decisions possible. So I think that it's perhaps there will be some good that comes from this time of reflection and introspection and research. Rachel, I'd love to just give you the last moment to share with us what you've thought from hearing all of this really wonderful science from Dr. Kong and understanding all the different options and therapies and processes, and maybe how it impacts how you feel and your list. So Rachel?
Yeah, thank you for letting me share. And, what was really poignant to me was looking at the photo from when Dr. Kong and I first met and did these visits, and, and realizing that was in 2016. At that point, I was about 10 months into the diagnosis, which, I look and see my chemo curls coming in. And I, at that point, I was still completely reeling from the diagnosis and from realizing that the life that I had pictured and the future I had pictured just wasn't going to happen that way. And to meet researchers like Dr. Kong in that moment, and to be involved with organizations like the American Cancer Society, and know that there are others who are taking up the charge and advocating for patients and going to work in labs every single day. I just want to, that gave me so much hope at that point. And, I sort of just want to put my arms around, 2016 me and tell that woman that, it was going to be okay. And that, because of the science that has been funded by the society, for the last 5 years, I have had a relatively normal life. I mean, I saw my daughter start high school. I saw my son hit his first home run. I'm home, every day after school. I had the opportunity last year to travel to Portugal and advocate globally for metastatic breast cancer research. And, because of all of the science and because of the dedication of donors to funding cancer research, my cancer is stable enough and my treatment is tolerable enough that I was able to start to think about a future and, what did I want that future to look like? And for me, I want to give other patients the future that I didn't dare dream of. I want them to have confidence in science and know that it's okay to dare to dream of a future. And the only way we're going to be able to give metastatic cancer patients a future is to invest in this research pipeline and to continue to support the researchers in coming out with new therapies and investigating new receptors and understanding how metastasis works and how it can be managed. I am on my third line treatment currently. The next drug that I will be on was only approved in February of this year. And so, hearing from Dr. Kang sort of the trajectory of the pipeline and how long some of these discoveries take to come from a basic scientific hypothesis in a lab or in a mouse model and then become, a therapeutic option to add to my list, just continues to drive home how important it is, the work that the society does and the importance of support of research. And, I need to be able to look in my children's faces when I reach the end of that list and tell them that I have done everything in my power to save their mother. And the funding of the research pipeline is the only thing that will allow me to do that. And I'm so grateful to all of the donors, to the society who believe in cancer research and, are able to offer a future to me and to patients like me through your support and through your philanthropy and generosity. So I would just say thank you, and I am grateful for you every single day.
Thank you so much, Rachel and Dr. Kong. And with that, I'll turn things back over to Lauren.
Wow. Well, Dr. Greer and Dr. Kong, thank you so much for sharing your expertise with us. And Rachel, thank you for sharing your absolutely incredible and uplifting story. And for those of you that joined us today, thank you so much to you as well. And we hope that you guys found this presentation valuable and inspiring. And we really thank you so very much for your ongoing support. Because of you and your generous donations, we're able to help fund research like this, but other cutting edge research, and we are just beyond grateful. If you would like to listen to this webinar again, please feel free to do so. It'll be posted to cancer.org within the next few days. And also, if you should have any questions or comments, please don't hesitate to call us. We're available at 1-888-837-3322 or by email at laurenashcraft.cancer.org. Also, if you're interested in hearing our podcast, Theory Lab, it's available at cancer.org/research podcast. Thank you all once again. And again, a big thank you to Dr. Kong, Dr. Greer, and Rachel. Have a wonderful afternoon.